I'm David Swain, the Laboratory Director of the Southeast Poultry Research Laboratory, which is part of the Agricultural Research Services U.S. National Poultry Research Center in Athens, Georgia. Our facility has a parallel research mission to Plum Island Research Center and the future NBAF. But instead of transboundary animal diseases of livestock, our mandate is transboundary diseases of poultry and other birds. We're an OIE collaborating center for research on emerging avian diseases and an FAO reference center for avian influenza and Newcastle disease. We've operated a biosafety level 3 enhanced facility and conducted research on 2 select agents, highly pathogenic avian influenza and Newcastle disease for the past 45 years, and we're undergoing a $158 million facility modernization project. We also have conducted emergency response research, which is the topic of today's session. On West Nile, we've worked with SARS, SARS-CoV-2, MERS, and 1918 influenza viruses. I also want to mention the other ARS location which conducts emergency response research on animal pathogens is the National Animal Disease Center in Ames, Iowa. And I'll spend the next few minutes kind of describing the types of research that we do at Southeast Poultry as well as at NADC in this emergency response category. So with emergency response research, it's really critical that we do this quickly. So it's really real-time, or at least near real-time, scientific solutions to address field control questions as they come up. And this we have experienced with Newcastle and avian influenza outbreaks in the United States. And I'll give you a couple of specific examples. For example, we've done agent inactivation under field conditions and in different organic and inorganic matrices to kind of inform control and prevention strategies, especially in cleaning, disinfection, and downtime on individual barns before they can be repopulated. We've also done a network analysis of molecular data that informs both the virus spread between farms as well as within barns within farms, and use this in helping companies as well as our regulatory agency, the Animal Plant Health Inspection Service, in developing intervention and mitigation strategies. We produce data on the efficacy of existing vaccines against new field strains so we can understand, will our current vaccines protect against these emerging strains? We've done rapid development of consensus sequence of protective epitopes for cassette replacement and non-replicating vaccines. An example of this is avian influenza in 2014, 2015, of which because of the regulatory flexibility of the Center for Veterinary Biologics, we were able to work with them and several companies in changing non-replicating vaccines epitopes to update those for potential vaccine bank use. We've also generated safety and limited efficacy data to accelerate the veterinary vaccine licensing of these products. So for example, the RNA particle vaccine was licensed for use in swine for H1 and H3 influenza. We were able, through this regulatory process and the data we generated, to get that relicensed for use in poultry for emergency application for an H5 influenza vaccine. We also have revalidated the sensitivity and specificity of avian influenza and Newcastle diagnostic assays, specifically the real-time PCR tests that are used by the National Animal Health Laboratory Network and the National Vet Services Laboratory against new outbreak viruses. We've also, as well as our sister laboratory at the National Animal Disease Center, conducted pathogenicity studies to determine what animal species are susceptible to emerging agents, determine their modes of transmission, and determine the likelihood of a reverse zoonosis. Probably the best example is the recent COVID-2 virus, COVID-19 virus, SARS-CoV-2, in which the National Animal Disease Center immediately did studies in white-tailed deer, cattle, and swine, and determining their susceptibility and potential for reverse zoonoses. And we at the Southeast Poultry Lab have done the same kinds of studies in chickens, turkeys, ducks, geese, and Coturnix quail. The other good example we had was from 2009 with the pandemic H1N1 virus, of which studies were done for emergency purposes in swine at the National Animal Disease Center. And the final point of emergency response research is the readiness to be prepared. And that is a critical piece for laboratories that must be able to run and switch their research portfolios to topics that are emerging or have emerged in outbreaks. For example, facilities have to be already constructed and operated and maintained in this BSL-3 or above type of containment system. Must have the manpower, the scientific expertise, and practice and the functionality of doing this type of research on an ongoing basis, and flexibility to be able to move from planned research studies to these emergency response studies. That's a good overview of emergency response research. Thank you.
Thank you so much, Dr. Swain. And now we'll turn it over to Dr. Jack Shear from the Animal Plant and Health Research Service.
I can't seem to get my camera going. Can you hear me though?
Yes, yes, we can hear you.
Okay, fine. I'm going to take a little bit of a different slant than Dr. Swain, and what Dr. Swain has talked about, I'll talk about a little bit because everything he's talked about hinges on disease outbreak response within 80% of experiences we've had. First of all, I would say this, that domestic disease serves as a platform and it does the groundwork and the testing that we need for when we get into a foreign animal disease outbreak. It's just that we have to scale up tremendously at a tremendous higher level. So outbreaks are areas where you're dealing with making decisions perhaps sometimes on 20% of the information. We have to have a situation where we have we can quickly do surveillance to find out where the disease is and where the disease isn't. And without testing, without the science of the testing, and having a surveillance plan to know what your diagnostics will tell you and what it will not tell you, you're sort of at the mercy of the disease. So we want to in any disease, we want to rapidly find where that disease is and where it isn't. Because, for instance, in the United States, every day we have, and it's been said, in the swine industry, we have up to 1 million pigs on the road every day going different places. In the state of Iowa, they talked about the fact that they have 4,000 to 5,000 trucks moving to and from throughout the state, hauling different commodities for the swine industry. So disease testing and surveillance becomes extremely important. The time span involved in that testing is critical. During the outbreak of Newcastle disease in California in 2002, 2003, we were trying to work with virus isolation to find the disease. And while that's a very good test, it just simply couldn't get us the answers quickly enough, especially if there were problems with the test. We were sometimes 7 to 10 days and even more behind the disease for a diagnosis of positive or negative before we could move on or react to and deal with the farm or the premises. So as we look forward in such diseases as foot and mouth disease and African swine fever, we want to be able to have diagnostics that are easy samples to get. And that I say because the industry, we want samples that will tell us quickly where the disease is, that are easy to retrieve, easy to, and quick, that are done within a day to 2 days, with that we have an answer so we can react and deal with the premises. And we want samples that are easy to and viable when they get to the laboratory. So all those things have to be incorporated into whatever test we're running and the science behind that test. There's a lot of discussion about pen-side testing, and that would be an optimum as we move forward with some of these fast-moving diseases. Pen-side testing is important. Why is it important? Well, when you think about the million hogs on the road every day, if we find where disease is and where disease isn't, then those other hogs that are not infected have to be able to move, and we have to be able to test them to move them. And that requires testing that not only is done fast, but is accurate, because we can't accept a test that only gives us 81% accuracy. State vets are not willing to see animals move on. They want 100% assurances that when they move those animals and we sign that certificate for them to move saying they're not infected and they're going from state X to state Y, that state Y is not receiving animals that perhaps have disease. So lends to the consistency and the mass testing ability, and that gets me into capacity. So lab capacity is extremely important. In the United States, we've developed a non-laboratory system. We've developed testing to increase our lab capacity and our surge capacity by bringing those laboratories on board. That science and that testing is ever moving forward. Some tests, as we know, have to be validated perhaps in the face of an outbreak. We just don't have the data, and we need that data, and we depend on our laboratories and the scientific scientists that are out there to help us get that validated, and people like Dr. Swain that have brought us forward in many cases with that situation. Vaccination is another question. We know that in some disease outbreaks, we're going to use vaccination, but then we also have to be able to quickly differentiate the vaccinated from the non-vaccinated, so that we're sure that, again, as we move animals based on testing, that if there's a test positive, that it's not an animal that's vaccinated, and that we do the correct things in that movement. So if you quantitatively add up all the movements that are going on during an outbreak and all the decisions that the outbreak managers have to make, diagnostics is at the heart of all of that. And the ability to have accurate tests that you're able to use quickly to get a quick answer and an accurate answer, so that you're making the right decisions in those movements, and you're not spreading disease based on a test that's not going to give you the correct answer. So we depend heavily, science goes heavily with disease outbreaks, and the work that is done in these laboratories is critical to us being able to make the right decisions and to move forward with those decisions, so that we're eradicating disease instead of making the wrong decisions and spreading the disease. I'll stop there, and I'll thank you very much for your time.
Thank you so much, Dr. Shear and Dr. Swain. We will now turn it over to our moderator, Dr. George Korch. George, if you wouldn't mind giving yourself a brief introduction, and then we will get started with our programming. Thank you so much.
Good morning. Thank you, Haley. Thank you, Dr. Swain and Dr. Shear. Without belaboring the point, we have 5 wonderful speakers today for you to hear from. I'm George Korch. I'm the director currently of the National Biodefense Analysis and Countermeasures Center. It's operated by the Battelle National Biodefense Institute for the Department of Homeland Security. So it's a biosafety level 3-4 facility where we look at mostly human pathogens, but we do have the ability to assist in agricultural pathogens as well. So without further ado, what I would love to do is start to introduce our very first speaker for the day. And we're delighted to have Dr. Kerstin Wernike, who studied veterinary medicine in Berlin, Germany, and has a doctoral degree in veterinary medicine from LMU in Munich, Germany. Board certified expert in veterinary virology, gained her habilitation in animal health and animal welfare at the University of Rostock. And so since 2016, Dr. Wernike is a senior scientist at the Friedrich-Loeffler Institute, Federal Research Institute for Animal Health in Germany. And she's the head of the National Reference Laboratories for Bovine Viral Diarrheal Mucosal Diseases and for Schmallenberg virus. In addition, she is the deputy head for the German National Reference Laboratory for the SARS-CoV-2 infection in animals, and she's held that since 2020. She has 11-year experience in working with different animal viruses under high containment conditions. Her main interests include emerging animal viruses, host virus interactions and immunoprophylaxis, generic characterization of viruses, and the development of diagnostic systems. And so without further ado, delighted to have Dr. Wernike discuss responding to novel emergent ruminant arboviruses.
Thank you for the introduction. I need to start my presentation. And also thanks to the organizers for inviting me. I will talk a little bit about ruminant arboviruses, and I've selected the Schmallenberg virus as an example. For those of you who are not familiar with that virus, it belongs to the Symposium group. It's related to Akabani virus. It infects predominantly ruminants and is transmitted by colicoides biting midges. SPV emerged in Central Europe in 2011 and spread since then. When the virus infects adult ruminants, you see either none or only very mild clinical disease. I will show a few slides after that one. The most important thing, we have a very short viremia, surprisingly short for a vector-borne disease of only up to 6 days. And the major problems come apparent when pregnant animals are infected very early in pregnancy. In cattle, about day 70 to day 170, it's shaded in red there. And sheep in the first to third month of gestation. After infection or in pregnancy, those malformed fetuses or some of the newborns are even alive, are born in a very high percentage of infected small ruminants and a smaller percentage of cattle. Most of those calves and lambs are born at term, and that's maybe the as we are in the emergency response research section, the biggest problem, because when the farmers and veterinarians see that clinical pictures, the virus circulated up to half a year ago. All the other pregnant animals in the farms may be already infected without any possibility to do something against it. As I've said, I will talk a little bit about the clinical signs in adult ruminants. Those are mean values of 6 animals each, 4 groups, and you see the rectal body temperature. There's a slight increase in some animals, decrease in others, but it's only for 1 day. When there's real fever, it's a very small subset of animals, and it's also gone after 1 day. The second clinical sign is a decrease in milk yield. That's a screenshot of an automatic milking system applied in one of the affected herds. As you can see here in September, there's a slight decrease of about 10% in the milk yield in that farm. That was only noticed in Germany in farms that have automated milking systems. In other farms where the milk yield is measured only once per month, it was not noticed. For one of the latter farms, I have an example on which I will show you how the virus spreads within an affected cattle farm. That farm is located in the area where the first sample originated. Here, the red dot is the farm I'm talking about. The blue farm is Schmalenberg. In that farm, there are about 60 cows, their female offspring, 2 breeding bulls. There was no introduction of animals from outside. We got samples from May 2011 to January 2012. In that period, no vaccination was applied against any disease, and no further veterinary treatments involving needles. We got the samples from every milking cow and analyzed them by ELISA. Every green field in that graph belongs to a negative serological result. As you can see, up to September 2011, the whole herd was seronegative. And then came the vector season and the introduction of Schmalenberg virus. Now everything that's red is a positive result, and I think it's very impressive. Schmalenberg virus spread across the complete herd. Every animal seroconverted within only 3 days. During that time, no clinical signs were recorded apart from fever in those 3 animals. This was only noticed because the farm was closely monitored for another disease, and body temperature was measured every day from the animals. Luckily, only very few animals were pregnant, and there were no malformation in the next winter and spring in that particular farm. we now leave the farm level and go to a larger scale. That's the Schmallenberg virus situation in Germany up to June 2012. Every blue dot belongs to an infected cattle holding, red dots to sheep holdings, and brown dots to goat holdings. And you see how fast the virus spread through half of Germany. And when we noticed those cases were predominantly malformed ruminants, it was too late to do anything because the animals were infected much earlier. In the next year, we again saw cases, but predominantly in the region that was not affected in the first year. And as we all know, vector-borne diseases do not respect country borders. The virus spread throughout whole Europe within only 2 years. There's the German-Dutch border region where everything started, and all dark gray countries reported Schmallenberg cases up to 2013 within only 2 years. All samples analyzed before 2011 were clearly negative in every country, so there's no indication of virus introduction before 2011. As we're in the section about emergency response, I will discuss a few response strategies. The first one, and maybe the easiest, is to wait for herd immunity acquired by natural infections. In a face-to-face meeting, I would now ask what you would expect what happens when we just wait and do nothing. Now I can show only a few maps of Germany. The maps were extracted from the German disease reporting system, as Schmallenberg virus is a reportable disease in Germany. Every dot represents a Schmallenberg virus detection or antibody detection before colostrum intake. The first map symbolizes the period from April 2015 to March 2016, covering one insect vector season, and the other maps the following years. Without further explanation, I think everyone sees there are dots in every year on every map on that slide. Even until last month, we had cases, and I guess it will not stop immediately now. So when you do nothing, wait for herd immunity, you basically have to live with the virus. Mainly because of the high replacement rates under European conditions. There are always enough seronegative animals for the midges to bite. The second possible response strategy could be the use of insecticides or repellents, but a German case control study provided no evidence for protection against Schmallenberg virus. Especially for Schmallenberg virus and the related viruses, one could consider seasonal mating in a way that the mating period is shifted in a way that the high risk period is in winter when there are no when there are no infected midges around. But that's sometimes hard to implement. And the last one, maybe the most important strategy, is vaccination. To show you a possible impact of vaccination, we will now switch viruses and go to blue tongue virus. We've heard about that virus and the clinics yesterday, so I will skip that. For my presentation, the most important point is that blue tongue virus infects the identical mammalian hosts, ruminants, and uses the same insect vectors as Schmallenberg virus, so the epidemiology is more or less comparable. Blue tongue virus serotype 8 was introduced into Central Europe in 2006. And to show you the spread from 2006 to 2007, I have a movie. Every yellow dot that comes up belongs to a cattle holding, and every orange dot to sheep holding. We are now watching the year 2006 as the virus spreads. Now we start in 2007. And maybe when the virus spreads on that map, you may notice that it's exactly the same pattern as we saw in Schmallenberg virus. And it's also very impressive how fast this vector-borne disease spread throughout the cattle and sheep population of Germany. In contrast to Schmallenberg virus, there was a compulsory vaccination campaign implemented against bluetongue virus in 2008 and in 2009, so following this picture, and that was switched to voluntary vaccination in 2010. Let's have a look on the impact of vaccination. That's again the cases reported to the disease reporting system. That's where vaccination, I need to pointer again. That's where vaccination started, and you see a significant increase in cases, and no detected case from April 2009 throughout the years 2009 and 2010. So that was a very efficient vaccination campaign. In addition to the reported cases, there was also monitoring for BTV genome in domestic ruminants. More than 33,000 samples were tested, and all of them were negative. So again, it was a very efficient outbreak control by the applied vaccination campaign. That leads me to the final slide, to the conclusions. The first one is novel viruses can emerge at any time and cause large-scale outbreaks. That does not apply only to ruminants or vector-borne diseases, as we've learned in the last 12 months with SARS-CoV-2. Also, vector-borne diseases can cause large-scale outbreaks and spread across the world. Here you can see a small vector in a large vector, a midge in a plane. Vector-borne diseases are very efficient at spreading. In the case of Schmallenberg virus, the emergency response was, the only possible response was to develop reliable diagnostic methods as fast as possible after virus detection, and to have good communication with veterinarians and animal owners, because when they saw the effect, they feared further malformed newborns, and they were not able to prevent that because their dams were already infected. And maybe my final thesis for that talk is that in ruminants, vector-borne diseases are difficult to control by means other than vaccination. And with that, I thank you for your attention.
Thank you, Dr. Wernike. We're now in a period where we are looking for questions from the audience. I'll start very quickly. So you've mentioned bluetongue, you've mentioned Schmallenberg. Do we know of any other zoonotic viruses that are related to Schmallenberg, for instance, as bunyaviruses or others that are transmitted by these same vectors, these chilicoid biting midges?
Yes, there's arboviruses in South America, but it causes only flu-like symptoms. And there's Shuni virus. We call it a kind of super Schmallenberg. It was detected back in the 1960s in Africa in a child with fever. Thereafter, only in midges or in ruminants. Only recently it spread to the Middle East, infects also horses, causes encephalitis in adult animals. And during the last year, it was detected in humans with encephalitis or meningitis in Southern Africa. So it could be a zoonotic relative of Schmallenberg virus.
Another quick question. What has been the overall economic impact, do you know, of Schmallenberg then, either in Germany or across the EU?
It was significant in the first year, but thereafter it was, for the farmer, because of the few malformed fetuses, but predominantly because of export restrictions that have been implemented in a few countries. It's still up to today, some countries require to test every semen batch before accepting semen from Germany or the other affected European countries. That's today the biggest issue.
And again, please submit your questions to Dr. Wernike by way of the chat room. Or I guess, Haley, if people want to ask directly using their speakers and their microphones and their cameras, that's great too. And last question, you mentioned the success of blue tongue viral vaccine. And I'm not familiar with the developments, current development of Schmallenberg virus vaccines. Are there vaccines on the horizon there too?
There are 3 vaccines licensed, one for France, one for Germany, one for the complete European market, but it's not applied. Because in a given area, we see a wave-like behavior of the virus. You have infections every 2 to 3 years. When the farmers see the effect, they are willing to vaccinate, but then it's too late. And 2 years or 3 years later, they have forgotten about it, and then we have the next wave.
Yeah. Okay, let's see. Into the Q's and A's. Any other questions? Let's see. Question: which insecticides from Gabriel Zollner and repellents have been tested to prevent Schmallenberg transmission in culicoides?
I cannot answer that question in detail. There were a few farms, and the insecticides and repellents that were routinely used in those herds were compared to neighboring herds that did not use any insecticides.
And another really great question from Dave Patton. Why did both Schmallenberg virus and blue tongue appear to start in the same locations in Europe?
It's the most cattle dense area in Europe, and the very dense in human population. And you have a lot of harbors, and you have Frankfurt as a great airport. You have Amsterdam, Rotterdam. A lot of incoming travel meets a large cattle population.
Another quick question from Luis Rodriguez. So what emergency research was done that led to the actual discovery of Schmallenberg virus?
I'm sorry? I understand only discovery of Schmallenberg virus.
What emergency research, there must have been emergency research that was done very quickly to identify Schmallenberg virus?
Next generation sequencing of samples from diseased cows. In Germany, cows with fever were selected. In the Netherlands, cows with diarrhea. Obviously, the blood from fever cow was a good choice. And by next generation sequencing, the virus was discovered, and within days, real-time PCRs developed and distributed among veterinary laboratories.
So I think in the interest of time, we will move on, but thank you so much for your questions. Thank you, Dr. Wernike. And we're going to move on to next presenter. And a slight change just in the schedule. We're going to hear from Dr. Lindsay Gabbert. Dr. Gabbert has worked as a research microbiologist with the U.S. Department of Homeland Security and the Science and Technology Division, and specifically at Plum Island Animal Disease Center for more than 10 years. She holds a graduate degree in Homeland Security with a specialization in agricultural, biosecurity, and food defense from Penn State University. And as a subject matter expert for the design of validation studies to assess viral inactivation, a lot of her work has been focused on decontamination and disinfection of select agent foreign animal diseases, including African swine fever and foot and mouth disease virus by means of chemical and physical conditions. So prior to her work with DHS, Lindsay worked for the U.S. Center for Disease Control and Prevention, studying the pathogenesis of arthropod-borne zoonotic viruses. So a delight to turn it over to Lindsay. And Lindsay, you may want to unmute your microphone.
There we go. Thank you. Thanks for that great introduction, and thank you from the Symposium organization team for inviting me to speak here today. Gonna pull up my slides. All right. Okay. Can everybody see that? I think so.
Yes, Lindsay, you look great. Thank you.
All right, thank you. Yeah, so good morning. Today I'm going to discuss a little bit about the DHS Target Advanced Development Team applied biosecurity projects that are currently ongoing at Plum Island, and hopefully the link from the laboratory-based studies to how they translate to increasing preparedness and emergency response will be evident by the end of my talk here. I'm going to move pretty quickly as I do have quite a few slides. So at this point in the symposium, I'm sure you're all familiar with Plum Island, has the overarching mission to protect U.S. livestock from foreign animal diseases. This occurs through collaboration between the 3 federal agencies, including Department of Homeland Security, USDA-ARS, and USDA-APHIS. A couple of years ago, in response to the increasing global threat and emergence of ASF, there was an interagency ASF task force formed at Plum Island. You've heard before about the development of emergency use vaccines, in addition to improving diagnostics and surveillance capabilities to enhance outbreak response. And today, what I'm going to talk with you about is the third leg of this task force, which is to evaluate disinfectants and decontamination methods to support outbreak response capabilities and disinfection efforts. And what we're terming these projects as the applied biosecurity projects. And really the overarching goal here is to generate laboratory scale data to fill in knowledge gaps that have been identified in the response and recovery sector. And really what we focus on is 2 main areas, which is virus elimination through cleaning and disinfection, and 3D or depopulation, disposal, and decontamination projects. All of the projects that we take on are stakeholder driven or in conjunction with other federal customers. What that means is we're not a for-fee testing laboratory. We don't test for products for commercial companies. So we really work with stakeholders, including the National Pork Board and APHIS, who have identified knowledge gaps in the field that we can generate data sets to help increase knowledge surrounding those. And the 2 things I'll be talking about today are evaluation of commercial disinfectants and composting as a carcass disposal method for ASF infected swine. So just to give a little bit of background on ASF virus itself and a couple of attributes that are important to these environmental inactivation experiments is that ASF is a large double-stranded DNA virus from the family Arboviruses. The DNA viral genome is going to presumably survive longer in the environment than your standard RNA virus. ASF has a very complex structure. So in general, we think of envelope viruses as being the easiest to kill microorganism. If you look at that pyramid in the bottom right, there's a hierarchy of susceptibility of microorganisms, and envelope viruses are on that easiest to kill bottom tier. However, ASF is quite complex in nature. It has 5 different layers. So the genome is surrounded by a core shell, and there's an inner capsid, an inner membrane, an outer capsid, and then an outer envelope. So that is something to take into account when we're looking at destruction of ASF in the environment. It has been shown to be viable for long periods of time in blood and tissues, especially at low temperatures, and is stable over a wide range of pH values. Like we heard in the talk yesterday, implementing sound biosecurity protocols on farms for producers is really an important part of controlling ASF. So this includes virus elimination through cleaning and disinfection, and also decontamination of mechanical fomites such as transport vehicles, clothing, and feed. So how do we know which chemicals are effective? In the laboratory, we primarily utilize what's called a quantitative carrier test to determine the efficacy of different test chemicals and physical decon measures. At the basic level, what a quantitative carrier test is a highly standardized and sometimes even globally harmonized test method that tightly controls the different experimental variables to be able to compare test chemicals. In general, it requires demonstration of a 4-log reduction in infectious virus titer to prove efficacy of a chemical. The table here just lists a couple of the different international test methods. We use the OECD test method, which requires 10 microliters of virus to be exposed to 50 microliters of the disinfectant. The virus is mixed in a standardized 3-part soil load to increase the challenge, and the disinfectant is diluted in hard water. And this is conducted on stainless steel test coupons. So just to give you a little visual of that, on the left here, we have our coupons with the inoculated virus drying. Then we apply our disinfectant for the particular contact time that we're investigating. We neutralize that disinfectant and then elute any remaining virus, which is titrated in a CPE-based TCID50 assay. There's a lot of pros to this method, including the small amount of virus used, the large neutralizer to disinfectant ratio, which decreases cytotoxicity in our cell lines, and really allows us to process a lot of replicates at one time to increase our statistical power. The con is that we need a really high titer of starting virus to demonstrate that 4-log reduction. So for ASF, for example, we have to concentrate our stocks to about 9 logs to start with. Give you an example of a recent project where these test methods were utilized. Back in 2019, we established a cooperative research and development agreement with the National Pork Board. And the purpose of this was really to evaluate commercially available liquid disinfectants that are in use by US pork producers. And the overarching issue here was that for the approved chemicals that are available against ASF right now, none of them are really readily available or being commonly used on farms. And so the goal was to look at chemicals that farmers and producers have in stock and already have protocols for use, and see if we could determine if they would inactivate ASF. So the Pork Board stakeholders identified 4 disinfectants from different chemical classes that were high priority. We tested them in the lab using that OECD method, and any products that were shown to be efficacious against ASF, but with no current registration or label claims, we reported that to APHIS Veterinary Services. This is just a snapshot of the data of the different test chemicals we looked at with a 10-minute exposure time on stainless steel. We use citric acid and Virkon as our benchmark chemicals, as they're already approved for use against ASF. And as you can see from the graph here, both of them did result in a higher than 4-log reduction in ASF. Of the remaining 4 chemicals, Viricid, Intervention, Tektrol, and Synergize, only one met that 4-log reduction and was considered efficacious by this method. And that data was then written up in a report and submitted to APHIS. And really the goal here is to increase the number of chemicals that are registered and approved for use before an outbreak of ASF were to hit American soil. So this is a table that's published by APHIS of their approved disinfectants. And really, we just want to increase the type of chemicals and the number of disinfectants that are available for use. All right, we're gonna switch modes completely here. This is our most recent project at Plum Island, and this really centers around evaluation of carcass disposal as a method for disposing of ASF infected swine mortality. And this was a gap that was identified by APHIS composting subject matter experts, as well as other composting subject matter experts around the country. Composting has been identified as a preferred disposal method for both routine and emergency management of animal mortalities. And this is opposed to your standard burning, deep burial, or rendering. So composting just as a method generally inactivates pathogens through increase in temperature in the composting windrows and just time. You can see in the pictures on the right here, examples of composting windrows, both in the field and inside of a barn. And I'm presuming these pictures are during the HPAI outbreak. And USDA has published a livestock mortality composting protocol back in 2017. And this was really the guide that we used to help us design the study to conduct in the lab. So our overarching goal was to provide evidence supporting composting's thermal and biological activities as a method of ASF inactivation in infected swine. We really wanted to replicate as closely as possible what somebody would do in the field. So we actually had to build a composting windrow and use different carbon materials to replicate that inside of our biocontainment facility. And opposed to measuring carcass decomposition, we were primarily interested in virus survival and detection of DNA over time. So this was definitely a challenge for us. There was a lot of operational, logistical, and biosafety challenges involved. Questions we had starting out were, how are we gonna procure these carbon materials and get them into containment, and how do we dispose of them? Is there going to be insect activity? What kind of heat's going to be generated? And what sort of biological samples should we take? So this picture here is actually an illustration that I made. It's kind of something for fun, but it was our original vision. How do we explain to the operations people what we would like to build inside of one of our animal rooms? So from our initial planning phase, we moved into the actual compost pile construction. This all took place in one of our animal rooms at Plum Island in the BSL-3 egg facility. You can see here we started off by construction of a wooden base, which we covered with plastic sheeting to try to contain any of the carbon materials or leachate that could come out of the compost pile. Around that, we also placed absorbent socks, which you can see better in the photo on the bottom right there. I like this first picture here because it gives almost a cross section of the different layers of the compost pile. So the bottom layer is a base of about 15 inches of municipal wood chips. That is covered by 4 to 6 inches of pine shavings. You can't see it, but inside there's 4 ASF infected pig carcasses, about 120 pounds each, which we covered with a horse manure and bedding mix as the core, and moisture was also added to that layer. That was all capped off by about 15 inches more of the pine shavings. And the finished compost windrow is in the bottom right photo there. The sample tags, you can see the colored plastic tags coming out of the pile, and I'll talk about those next. So like I said, we had 4 ASF infected pigs that we utilized for this study. We decided to use the spleen as our primary sample material that we were going to look at throughout the duration. So a spleen from each pig was harvested directly from the abdominal, removing it directly from the abdominal cavity and sectioned into about 30 pieces per pig. This was then added to our sampling bags with compost. There was 24 bags per pig. Those were reinserted back into the abdominal cavity so that it was as close to the animal inside the compost pile as possible. We had 3 different types of temperature monitoring throughout the experiment, really to make sure that we got some sort of good temperature data, as this was really important variable to show that our compost pile was working effectively. So we read the temperature of the compost pile daily manually with 36-inch long compost thermometers. We also used thermocouple probes, which are essentially long wires that went to a data logger in a window so that we could look at the temperature without having to enter the room. And we also used hobo data loggers, which were buried within the compost pile. There was 4 of those, and those took temperature readings every 10 minutes for the duration of the experiment. So the total experiment lasted 37 days. We collected 3 of the bag spleen samples from each of the animals at days 0, 1, 3, 5, 7, 10, 14, 21, and 28. At day 37, we opened up the compost pile to review the carcass remains and also take sentinel samples of the carbon materials. And the photo here, you can see our sampling bags. The material was so hot that steam was forming on our samples when we collected them. So just a couple of result slides here, and then I'll wrap it up. We were really happy with the temperatures obtained in our compost pile. Between days 4 and 5, the temperature rose to 130 degrees Fahrenheit, 54 degrees Celsius, and it stayed there for over 20 days. Our peak temps were seen on days 6 to 10 and were approximately 150 to 160 degrees Fahrenheit. And one of the take homes that we noticed was that our manual temperature reads did match very nicely with the data obtained from our buried data loggers. Preliminary laboratory results. We have analyzed all of these samples via RT-PCR for detection of viral DNA. We found that all of the spleen samples have been positive through day 28. All of the tissue samples collected at the end of the experiment from the decomposed carcasses, including bone marrow, muscle, and ear, were positive for virus DNA. The highest concentrations were seen in bone marrow. And sentinel carbon materials, we took 16 random samples and 14 of those were also positive. So this suggests that composting in the field, even if you do have an effective composting process, you most likely will see positive virus DNA samples at the end of your experiment. We've also looked for live ASF through virus isolation. Our starting titers in all of our samples were about 5.7 logs. We've detected infectious virus on days 0 and 1. 1 out of 12 samples was infectious on day 3. And between days 5 and 10, we could not detect any live virus in any of our samples. So that coincides nicely with that increase in compost windrow temperature. We're currently evaluating the bone marrow samples right now. And thus far, we have not found any live virus after 37 days in the bone marrow samples. So that's all I have for you today. Thank you for listening, and I'd be happy to entertain any questions.
Thank you very, very much, Dr. Gabbert, for that outstanding presentation and really fascinating work. A few questions that are coming on in. One from Doug Yvonne. Basically, the question is, does the disinfection on the surfaces that you mentioned earlier with the coupons, does that follow a first order kinetics?
I'm assuming by first order kinetics, they're referring to, looking at concentration versus contact time.
I would imagine.
So yeah, I think it really depends on the test chemical. We've done a lot of experiments in our lab, both with FMD and ASF using Virkon, as that's the primary disinfectant we use in the laboratory. And it's really hard sometimes to measure the kinetics because your virus is inactivated within 30 seconds. So looking at longer contact times or different concentrations sometimes isn't possible, but yes, in general, I would say that either increasing the contact time or the concentration, you'll see inactivation, kill curves proceed accordingly.
Great. And same questioner basically wanted to know how you generated or where were the African swine fever pigs acquired from?
So I'm happy to say they were actually recycled from another ongoing vaccine efficacy study that USDA was conducting. So in a vaccine study, you obviously have control animals that you have to demonstrate become sick. So we just took those at the end of the study and used them in our composting experiment.
A quick question, I think, related to the question of kinetics. First of all, you got a compliment on your nice composting artwork. I was very impressed with that as well.
Thank you.
Do you ever look at varying the contact times, concentration with disinfectants for inactivation curves? Or are you pretty much just evaluating the manufacturer's recommendations?
For certain experiments, yes, we are looking for kind of the sweet spot, what concentration and contact time is effective. For that project specifically, because there are such strict regulations put in effect by the Environmental Protection Agency on how a producer can use a product, they're only allowed to use it according to the instructions that are listed on the product label. We wanted to keep our testing conditions really tailored exactly to what was already listed on the product label, so that, you know, if an outbreak occurred, the products could be used in the same way that they currently are being used. We
just did the same with coronavirus SARS-2 experiments in our lab using the manufacturer's recommended contact times as well for the same reasons. Dr. Swain asks, how ultimately did you dispose or remove the windrow materials from the BSL-3? Did you have to incinerate, render, autoclave, or was it, did you test and find no viable viruses in the windrow material?
Yeah, so everything at the end of the study is technically considered select agent material. And luckily our permit with New York State allowed all of the carbon materials to fall under our facility incineration permit. So the carbon materials were all containerized in large garbage cans and sealed and then incinerated.
Quick question before we wrap up here in just a few minutes from Jürgen Richt. Will different seasonal conditions affect composting itself?
Yes, I would say definitely that it would. I'm not a composting expert, but I really learned through this experiment that it's definitely an art as far as your airflow and moisture levels and environmental conditions. We conducted this at room temperature, but I could see if you're doing this in the winter, it probably just slows everything down as far as your pile getting to the correct temperature and maintaining that temperature for the required length of time.
Just a quick question, because you mentioned RT-PCR, or did you look at whole sequencing? This is a question for me. Did you see any DNA degradation at all?
Yes, so we did. So when we're looking specifically at those spleen samples where we took them over the entire time span of the study, we're seeing much higher levels at the beginning of the study versus at the end. But I think, you know, if this one, I guess, surveillance or diagnostic method, if this occurred in the field, people would want to sample the compost pile and be able to use a PCR negative to confirm that the experiment worked, and we're really just not seeing that as a possibility at this point.
That sort of answers another question from Amresh Das with regard to does temperature alone cause the inactivation, or are there other chemical reactions in the compost that we're unaware of? So is it a function of just a temperature, or are there other breakdown products as a function of tissue lysis?
Yeah, I'm sure there's a lot of other microbial processes occurring, especially just as a carcass decomposes on its own without that added heat. So there's different microorganisms, there's different decomposition processes, there's pH changes within the carcass that are all occurring simultaneously. for us, unfortunately, we just have that temperature variable that we can monitor, and everything else is natural. So.
And the last question before we thank you very much from Vivian O'Donnell. You mentioned 10 microliters of virus treated with a 50 microliter disinfectant. And can you expand on how the samples were treated actually to concentrate when working with the high virus titers? Now, I'm not sure if that is a question of how you concentrated virus stock to begin with.
Yeah, hi Vivian. So we actually produce large stocks of the ASF BA71V, and we concentrated those through PEG precipitation. And the reason why this is necessary is that previous decon studies conducted by Peter Krug have shown that ASF titers rapidly decrease through desiccation when you're drying it on surfaces. And so in order to demonstrate that 4 log reduction, we really need to have a high starting titer, just because we lose 3 to 4 logs just through drying. So that's giving us a 6 log starting point to work with in these experiments.
Great. Well, thank you so much, Dr. Gabbert, for this fantastic presentation. We will be moving on now. Again, we had mentioned there was a slight change in the presentation order. So we are going to next bring to the podium Dr. John Hardham. Dr. Hardham is the director of the Zoetis Center for Transboundary and Emerging Diseases and the Veterinary Medicine Research Development Emerging Infectious Disease Programs at Zoetis. He earned his Bachelor of Science from Penn State University and his master's and doctorate from the University of North Carolina. And following his postdoc fellowship with the University of Texas Health Center at Houston, he joined Pfizer Animal Health in 1999, and he continued following the transition of that organization to Zoetis in 2013. Dr. Hardham has led multiple project teams in developing vaccines, biopharmaceuticals for both livestock and companion animals. In addition to working for Zoetis, Dr. Hardham, and I know this because we served together, is a captain in the United States Navy, where he specializes in the chemical and biological defense research programs. And he's served on a variety of committees for the National Academy of Sciences. He's currently assigned in his role in the Navy to the Naval Expeditionary Combat Command, and he's detailed to the Defense Intelligence Agency, the National Centers for Medical Intelligence. So delight to welcome John Hardham to the podium.
Thank you, George, for the introduction, and thank you to the organizers for inviting me to present today. So I will be presenting an industry perspective on dealing with transboundary and emerging diseases. If I can get my slides to show here. There they go. So I am the director of the Zoetis Center for Transboundary and Emerging Diseases, and this is the organization within Zoetis that is charged with tracking and surveilling and developing products to mitigate the impacts of transboundary and emerging diseases. So from an industrial perspective, we have a significant interest in assisting our customers, be they producers or private owners of companion animals, to mitigate the impact of transboundary and emerging diseases. These diseases can have a direct impact on animal health and well-being, whether they're production animals or customer-owned animals. They can also have significant impacts economically in various regions around the world, in addition to indirect impacts on quality of life and One Health issues regarding zoonotic transfer of diseases back to humans. In addition, they can impact the health of humans due to impacts on protein supply. As I mentioned, in Zoetis, we have a Center for Transboundary and Emerging Diseases. This is our organization that ties together all the different aspects of our company, from our R&D organization to manufacturing, to our US and international business operations, legal, communications, public affairs, etc., with the goal to help expedite our processes and be as efficient as possible in dealing with transboundary and emerging diseases. So we view the ability to mitigate the impacts of transboundary and emerging diseases as a continuum of care. And we look to provide products and services across that continuum of care in what we call predict, prevent, detect, and treat. If you look at the predict component, our center surveils diseases, conducting active and passive biosurveillance. In Africa, we have a partnership with the Bill and Melinda Gates Foundation, referred to as ALPHA, that we're able to obtain surveillance data from that continent through our reference labs, through our field force, and through our genetics business as well. Of course, in prevent, we actively develop vaccines for transboundary and emerging diseases to assist customers, producers, and governments to deal with these diseases. A couple examples there. We have a licensed classical swine fever vaccine called Suvaxyn CSF, and we're actively developing vaccines for foot and mouth disease, African swine fever, and COVID-19. From the detect component, we look at providing diagnostics, either pen-side or laboratory-based, PCR, lateral flow, or ELISAs. And then, of course, from the therapeutics aspect, we look at either pharmaceuticals and small molecules or biopharmaceuticals, such as monoclonal antibody therapeutics. So in looking at a couple of those key transboundary and emerging diseases, classical swine fever, African swine fever, foot and mouth disease, and earlier this week, Elizabeth Reeder from Plum Island presented some of the work that we've been doing in collaboration with her group on the foot and mouth disease vaccine platform, FMD LL3B3D. We have, as I mentioned, a licensed Suvaxyn CSF vaccine. It's licensed in Europe and has a permit for distribution and sale in the US. But we're actively working with African swine fever, Georgia, live attenuated virus vaccines. And what I'd like to actually talk about the rest of my talk is responding to the COVID-19 pandemic. I figured that would be a bit of a timely topic to discuss further for emergency response. So when the first cases of veterinary COVID-19 were reported, we set out to initiate a program for vaccines, diagnostics, and therapeutics. Given that we have a number of coronavirus vaccines licensed in various species, we first sought to develop an antigen. And for SARS-CoV-2, we looked at the spike protein. It's graphically shown on the left. And what we expressed is just the ectodomain of the spike protein. And we utilized a eukaryotic expression system that we've utilized for other vaccine antigens as well as monoclonal antibody therapeutics. And we expressed this soluble trimeric spike ectodomain. We incorporated a few extra features in order to lock this into a trimeric conformation, as well as also locking it into the pre-fusion conformation. Being a metastable protein with both a pre and post-fusion conformation, we were in need of keeping it in the pre-fusion conformation, and as such, we generate about 14 to 40-fold higher levels of neutralizing antibodies. And when you look at a transmission electron micrograph of our spike antigen, you can see that we have a nice cone-shaped structure with an elongated tail that perfectly mimics that of the crystal structure of the pre-fusion protein. And when you zoom in on that, you can see we have a perfect overlay with the crystal structure of the protein. So this is the antigen that we've utilized to further develop the experimental SARS-CoV-2 spike-based vaccine, as well as the target for some of our serological assays. Now, following the initial reports in companion animals, we at first conducted a adjuvant selection study in dogs and cats. This is an example of the cat data, and this was presented at the World One Health Congress last fall. And we utilized two different adjuvant systems with a 2-dose vaccine series, 1 ml given subcutaneously. We collected blood and analyzed the serologic response using 4 different serologic assays. The first 2 are viral serum neutralization assays. This first one was conducted at the University of Tennessee, and they developed this assay for use in evaluating human vaccines. The second is a surrogate lentivirus system in which the lentivirus expresses the spike protein and binds to a host cell expressing the ACE2 receptor. And we have a number of these lentiviruses available expressing the wild type and variant spikes. And the last 2 are internally developed serologic assays using the spike protein as the target. And the first is a lateral flow assay, and the second is a typical ELISA assay. And this is an example of the data from that study from the University of Tennessee, the serum neutralization of the wild type SARS-CoV-2 virus, and red being the control, orange being the adjuvant system 1, and green being the adjuvant system 2. And pre-vaccination, all animals were negative. Post-vaccination, we got a mixed moderate response of serum neutralizing titers. And following the second vaccination, we got a very high titer serum neutralizing response. And you'll notice this is flat across the top because we maxed out their assay at the 1:2018 dilution. This was the maximum dilution that they had needed in establishing this model for use with the human vaccines. So we met or exceeded that threshold. Now, that initial data was done with companion animals. However, when the mink industry began to be affected, we pivoted over and started assessing our vaccine performance in mink models. We've conducted a number of laboratory and field safety studies of this vaccine in mink. And when the USDA issued the memorandum shown here, we are coordinating and working with the USDA in establishing the regulatory path toward a US conditional license to make a vaccine available to that industry. Earlier this year, we were approached by the San Diego Zoo after they had an outbreak in their gorilla troop to see if we could provide experimental vaccine to them to help assist in protecting their other great apes. We did happen to have a limited supply of experimental vaccine left over from one of our mink studies, and we were fortunate to be able to help the San Diego Zoo with that in collaboration with USDA CVB, who acted extremely quickly in helping to assist the zoo in protecting their other animals. So we were able to provide a limited number of doses to them, and they were able to vaccinate the gorillas, chimpanzees, and bonobo troops. In addition, of course, to great apes, there are a number of other animal species that are susceptible to COVID-19. Primates, there's been a number of reports of big cats around the globe that have been infected. Ungulates, such as the white-tailed deer, small carnivores, mustelids, of course, like minks, and perhaps even camelids are predicted to be susceptible. So in addition to responding to the outbreak in the veterinary community with the vaccine that's under development now, we've also developed a PCR assay to assist in identification of animals that are COVID positive. This PCR assay is very similar in nature to the human PCR assay in targeting 2 locations in the N gene. This is available at our reference laboratory, of course, as is very similar to many other organizations and veterinary diagnostic labs that have PCR tests. And we've also developed a lateral flow assay that I showed you earlier. This utilizes a cassette with the spike protein in order to detect the antibodies to SARS-CoV-2. Uses serum, plasma, or whole blood. In a typical lateral flow assay, 1 drop followed by 2 chase solution. After 10 minutes, you can visualize the presence or absence of a band. And we also have a lateral flow assay optical reader that can measure the density of this band to provide a numerical readout. And this assay has been validated at this point for dogs, cats, and mink. Now, in the last couple moments, just like to mention the variant viruses that have arisen. Of course, the wild type virus has been utilized for much of the susceptibility studies in various animal species. The variant viruses that have since evolved appear to have increased infectivity, transmissibility, and grow to higher titers in mammalian hosts. So it'll be important to assess not only what impact the wild type viruses have on animal species and their ability to spread, but also what these variant viruses can do. And there have been a couple papers looking at the prevalence of wild type SARS-CoV-2 in companion animals and other species. I'd like to point out this paper that was just very recently published from the CDC's One Health group, looking at seropositivity in pets, in persons or in households with laboratory confirmed SARS-CoV-2. And in this paper, they saw about a 12% positivity in dogs and 31% in cats. And then the second paper on Ferguson et al. Looked at what the impact is of B117 infection in dogs and cats. And interestingly, they found unusual clinical manifestations in dogs and cats with severe cardiac abnormalities with or without respiratory signs. So with that, I'd like to go ahead and close.
Thank you very much, Dr. Hardham, for the fantastic presentation and the products that you're working on, especially companion communities, but also for other veterinary communities out there, the zoo community, et cetera, and make it as well. So question first from Jeffrey Dennis. Do you follow any international standards when developing and validating your internal methods? And if so, which ones? And I'm not sure if that relates to the diagnostics or the vaccines, but a general question.
So for vaccines in the United States, I'll give the example, we are regulated by the USDA-CVB. So we follow all appropriate requirements set out by the USDA-CVB for safety and potency of the vaccines that we develop.
Quick question that I had from my own, and maybe you can or cannot answer this one because it's fascinating to see that you stabilized the spike trimer into the prefusion state. Were you using the proline kind of methodologies for stabilization or another methodology? And I don't know if that's trade secret or what, but just a quick question.
Yeah, we have licensed intellectual property for the process to stabilize that spike protein into the prefusion conformation.
Okay, well, thank you. Good enough. How quickly, from William Wilson, can you adapt SARS-CoV-2 spike vaccines to the vaccine variants if you need to do that?
So the system we utilize is literally a platform system for us, utilizing a eukaryotic expression platform. The time frame to turn around a new construct, obviously we can synthesize directly, clone it into our system, and then create a stable eukaryotic cell line. That process takes roughly a month or so. And then, of course, we'd have to generate that into a master seed, conduct all the master seed testing, and then the regulatory process is really the longest component to get a variant virus out as a vaccine.
Another question, this is from Randall Wenzel, and that was the mink vaccine used in the zoo outbreak? So it's the same also vaccine that you used to develop for felines.
Yeah, it is the same vaccine. The adjuvant system, the components of that adjuvant system have been used in a variety of other licensed veterinary products. And the key component of that adjuvant system is an immunomodulator. The receptor for that immunomodulator is expressed in a very wide variety of mammalian species. So it was able to be used across the different species.
A quick question also, this is from me. What method of a challenge did you use for the animals?
So the data that I showed there was just serum neutralization. We have conducted a challenge study in collaboration with Kansas State University. And we are looking at developing a challenge model in one of our European facilities for future regulatory testing.
Very good. Yes, I sort of jumped over the basis of the question itself. Chris Ora would just quickly like to know a brief update on the progress for developing your live attenuated African swine fever virus vaccine.
So we had publicly disclosed that we'd licensed the 3 of the USDA-ARS strains. We've transferred those to a overseas facility of ours where we can conduct the advanced development work. Obviously, with those strains, many of the data that have been generated and published on thus far with those strains and other live attenuated ASF strains has been done using primary cells. So from a manufacturing perspective, one of the first things that would need to be done is to identify and transition to a manufacturing friendly cell line. So we're conducting that type of work, as well as working with some of our key partners in Europe to conduct safety and efficacy studies.
Quick question back from Luis Rodriguez with regard, I think, for the SARS-CoV-2 vaccine. How stable is the vaccine? Does it need to be refrigerated? So
the formulation that we use is a liquid ready-to-use formulation, and it is refrigerated.
Very good. Simon Sang asks, can you comment on SARS-CoV-2 or COVID-19 susceptibility in livestock species as well?
So there has been a number of both in silico and laboratory studies conducted looking at the susceptibility. From the in silico perspective, you look at the ability of the spike protein's receptor binding motif to bind to the H2 receptor and the strength of that amino acid to amino acid binding. And in those, it's predicted a wide variety of animal species able to be susceptible. Cattle, for example, are predicted to be able to be susceptible. Swine, a bit less so, kind of on the borderline. Of course, ACE2 is only one of the binding capabilities for SARS-CoV-2. There are some others. So it's not fully predictive. It's just suggestive. From the laboratory perspective, most all these studies have been done with wild type SARS-CoV-2 and have been done in younger animals. Just like humans, we know that younger animals are a bit more resistant to SARS-CoV-2 than geriatrics. Of course, it's very difficult to have geriatric animals in studies such as this, so we have to use what we are able to. And in cattle, they were able to infect varying degrees of symptoms and shed virus. Swine appeared a bit more resistant to infection. A number of other zoologic species have all been shown to be able to be infected either in the lab or naturally, including that list that I showed: great apes, big cats, small primates, white-tailed deer, of course, mink and ferrets. And then actually this past weekend, the Georgia Aquarium had announced an outbreak in their North American river otters.
The last question, I encourage all of the speakers to also look in the Q&As, because there may be a question or two posed to you that we didn't get to that you can answer live, or type your answers out to. And the last one, although we won't ask it for your particular, for right now, if you're answering right away, because we need to move on, is for Margaret Allen. And I think you touched on a little bit, are there risks associated with the U.S. exotic pet trade? And again, that would be species dependent in a sense, but if you could answer Margaret's question through the chat room as well, that'd be great.
I'll go have a look at that one.
Thanks very much, John,
Thanks,
for
George.
fantastic presentation. And now we're going to move on to the fourth and fifth speakers for the morning. So delighted to introduce both of them. I'll do it together, and then they can swap off during their presentation. So Dr. Mia Torchetti joined the NBSL as the Diagnostic Virology Laboratory Section Head for Avian Diseases in 2013. And in late 2019, she was selected as the DVL Director, so expanding horizons to new viral pathogens, including SARS-CoV-2. She has a veterinary degree and master's in epidemiology from Colorado State University, and a PhD in postdoctoral work with ARS in Athens, Georgia. She has both national and international disease and response experience, particularly for avian influenza and for Newcastle's disease virus. Since joining the DVL, the opportunities to strengthen public and animal health relations and responses are critical as they help us to discover new ways to pull together the lessons learned from the largest animal health event in the U.S. caused by the introduction of the Eurasian H5 high-path avian influenza at the end of 2014, helped really to improve our collaboration and cooperation. Dr. Christina Loiacono joined the NBSL in 2005 as a vet pathologist and focused on foreign animal disease diagnostics and on education. She transitioned to the NBSL's National Animal Health Laboratory Network as the Associate Coordinator in 2011 and was selected to be the Network Coordinator in 2017. She currently leads a network of 60 federal, state, and university-associated animal diagnostic laboratories across the U.S., providing surveillance and outbreak response testing for 14 of the diseases of high consequence. And so I would be delighted to turn this over to Dr. Torchetti.
Thank you very much, and we really greatly appreciate the invitation to present with this group today. It's been a very enlightening session so far. I've been delighted to hear from the other speakers, and we hope this can contribute a little bit more information to the context of what we're doing for emerging diseases overall. Go ahead, next slide, please. Well, actually, maybe I should just state that those photos are of the campus here that comprises 3 different units: the Center for Veterinary Biologics, the National Animal Center for Health for ARS, and also the National Veterinary Services Labs, which the Diagnostic Virology Unit is a part of. Next slide. So what is diagnostic virology? We're a little bit different within the context of the National Veterinary Services Laboratories units in that we have responsibility for a larger portfolio of diseases. We look across multiple species. We're looking at several different viral pathogens. We function, we have select agent registered space within our BSL-3 space that we conduct the testing in. And of course, we've been tasked along with FADL to be on the ready for emerging diseases. And when SARS-CoV-2 started to be diagnosed in people, we were rapid to make sure that we were ready to test animals when needed. Next slide. So as with any other disease, there's a couple of different aspects that we need to consider. And one is the reporting aspect. And that is whether that's a state reporting level, the reporting at public health level, a national reporting level, and an international reporting level. In order for us to have the criteria needed for us to confidently and accurately report, a case definition is required. I believe we've modified this case or evolved this case definition over time. We want to make sure that we're capturing the current knowledge at the time, and we want to make sure that we're distinguishing a case that we consider high confidence for reporting at an international level is not the same as was that animal exposed. And so I think that that's really, really important distinction here, particularly early on. We wanted to have very, very high confidence that what we were seeing represented infection in those species. So I'll leave it to you to read some of our case definition. It is available on the publicly on a website. Bottom line is that we've defined what would be a suspect, a presumptive, and a confirmed positive case. And I know Dr. Loiacono is going to be speaking a little bit more about, from a reporting side, how we're trying to apply these definitions to data that is coming into us and being able to represent what's happening in the US as far as testing in animals. Next slide. So as you can imagine, standing up testing, we're early on in the disease. We're not sure what criteria will be used to, or what even samples are going to be good because we're just starting out. So we really wanted to look across. We wanted to make sure we could look at swabs and tissue, virus recovery, and then does the animal mount an immune response at the end. And fortunately for the majority of our early cases, actually majority of all of our cases, we have been able to look across all three of these sample types. Next slide. For our real-time PCR, initially we were wanting to make sure that we could gauge sensitivity and specificity, particularly for when we're looking across different animal pathogens. We were very, very fortunate to be able to leverage the work that public health had already done. We embraced the assays that CDC had put out, and we went ahead because that would give us a quick and early start to this, to evaluate across looking at specificity. Let's look at samples that we know are coronavirus positive across different species so that we can make sure that this test does not cross-detect those. And so we worked on that. However, we're not a companion animal lab. We're not a dog and cat lab. And so we're really, really grateful to be able to have engaged with our National Animal Health Laboratory Network partners, with USGS, to make sure that we had a complement of different types of species and samples that we could look at. And so we were also able to get a panel of coronavirus positive samples for typical cat and dog pathogens, as well as even serum and some samples for other mink pathogens. The other thing that we wanted to do early on was make sure that we weren't impacting or that we could conduct testing without impacting reagents required for public health use. And that was a very important aspect. And I know many of our non-lab partners, private lab partners probably face the same conundrum. Next slide. So how, again, we're looking at, we are a diagnostic lab, not a research lab predominantly. So we want to make sure that we are confidently reporting out what we believe is a true infection. And so looking at the combination of PCR and sequencing and being able to get an answer, say, within 24 hours versus a few days or a couple of weeks when we're looking at full genome, we wanted to make sure that we were targeting our efforts on something that could give us the most rapid answer. And Sanger is one of those tools that really helps get us there. This allowed us to look across the nucleocapsid, which is where our PCR targets are, but also 2 other gene targets, and beyond if we needed to. We looked and settled on these three gene targets to make sure that we were detecting viral genome outside of the PCR region. Next slide. For full genome, we've looked at a variety of methods. And again, it's just been tremendous to be able to look across so many different people working on this virus. Full genome protocols were rapidly reported and shared, which was really just a wonderful thing to have happened. And we evaluated several of them. Currently, we have basically settled on, we have one that we use when we need a more rapid turnaround. So we use the S5-IN torrent for a more rapid turnaround that will almost always give us some usable data and often full genomes across to be able to leverage. Then we also have, we've been using the CDC protocol for full genome on MySeq as well. Next slide. So just touch a little bit on virus isolation. Early on, we discovered that even samples with strong CTs we weren't necessarily recovering from. And generally speaking, it was only those samples that had CTs less than or equal 25 that were successful for virus recovery. Even many of those were not. But with a little bit of tweaking and optimizations, we tried some different cell lines according to input from others. But we have now kind of settled back within our own in-house cell line, and we have been able to recover for up to CTs of 30. But again, this is a little bit of a challenge for us from the standpoint of how do we have a protocol that we can say, yes, there's viable virus there, or no, there's not. And I harken back to the earlier presentation, looking at ASF from Dr. Gabbert. We know we can detect viral nucleic acid after virus viral elimination or inactivation procedures have happened. So how do we help build that confidence that we no longer have viable virus there? And that's something we're working on with our partners there at Southeast Poultry. And I know that others are working on it as well. And I sure hope that we come with a good solution for that. Partners at Southeast Poultry are also working on looking at how to perhaps modify our cell lines to make them a little bit more permissible to this particular pathogen. And I just provided there an example of the different species, the CTs of the diagnostic samples and the CT of the VI positive samples to give you an idea of, we certainly have a much broader range on the molecular detection side than we do on recovery of viable virus. And the otters, just to mention the otters, I believe those were Asian small clawed otters at the Georgia Aquarium. Needless to say, we might expect other otters to be susceptible as well. All righty, next slide. So virus neutralization, it was very important for us to be able to have an approach for detection of antibody that was species agnostic. We really didn't have time, really, quite honestly, to be able to stand up something that would be, working for every species. So we went with classical virus neutralization. Again, we conducted analytical sensitivity and specificity work using known serum of known status. And again, our NALM partners and private lab partners offered to help us with the dog and cat samples so that we could make sure that we're getting a good look across these this assay. Currently, we've tested over 770 serum from cats and dogs alone at NBSL, with approximately 30% being positive for positive by virus neutralization. These are of our diagnostic samples coming in. We've tested in total about over 1,200 serum samples from a variety of species, and we do have a high confidence We do have a cutoff that we leverage. So basically titers of 32 or greater are considered positive in absence of other positive findings. And we will continue to evaluate the data as we go forward and identify other species that have been affected. We're also fortunate with our ARS colleagues that have been doing infectivity studies for them to share materials from their experiments so that we can continue to evaluate what we're looking at. We've also looked at a commercial surrogate virus neutralizing ELISA that has an EUA now, and it has performed very well compared to our VN as a screening tool, and we'll continue to look at that data. Next slide. So this is wanted to give you a sense of the number of samples we've received, and actually I do believe this is all our samples, not just swabs that we have received. And you can see that, you know, by far and away, mink we've received and tested the mostly mink samples. I did want to highlight that that is largely due to partnerships with CDC and the Wildlife Services, looking at testing in and around positive mink farms. So just wanted to highlight that. And then you can see that generally speaking, we do have this kind of other group that comprises several other species, the majority of which have been negative, some which have been confirmed positive, which we'll talk about here in just a minute. Next slide, please. Also wanted to give take a moment to highlight a tool that we're using here at NBSL. It has been used for many years, was developed for the use of, or for looking at TB. And it has, we initially started trying this out with avian influenza. So during the high path outbreak, we used it during the Newcastle outbreak as well. So we're really starting to see its application to viruses. And where this tool can really shine is there is no algorithm to bias it. So when we look at phylogenetic analyses and or even network analyses that we've conducted, we've used both of those tools quite heavily. There's always that algorithm behind. If there's something off with the algorithm, what we're going to see may not be completely accurate. When we're able to use these things in tandem or add other tools, it really, really helps raise our confidence in what we're finding. And these, for VSNP, we have been, okay, good. We are looking at using this tool to help us in addition to our other tools that we normally use. The beauty of this one too is our bioinformatics team were able to identify, you know, when we look at these tables, we're looking at only differences. We can identify the genome location over the top and how that single nucleotide polymorphism affects at the amino acid level at the bottom. This has been an evolving tool, and it's been really helpful for us to be able to identify SNPs of interest. Next slide. And again, this is just a very low resolution phylogeny. I did want to highlight that at least in the US, nearly all of the animal viruses that we have detected fall into Gisaid clade G, GHGR, which is defined by that D614G substitution. And so basically we don't think that that holds any particular relevance for the animals themselves, but rather representing more of what's currently circulating in humans and spilling over into animals. But that is something we will continue to monitor very closely. I just highlighted on this slide, as was previously noted, we are starting to see variants of concern, a variant of concern, B117, spill over into animals, both pets and zoologic animals, as well as some variants of interest. And we will continue to monitor these over time. And we're a little bit trying to narrow what we're trying to get that full genome on, but we will attempt anything which is of high concern, but we want to be really rigorous with the data approach that we're taking with these. Next slide, I think that's about it for me. And just wanted to mention, we do try to do announcements. USDA conducts announcements for any new species detected, so look out for the one for the otters. And I will then now hand it over to Dr. Loiacono, who will continue on this presentation. Thank you.
Great, thanks, Mia. Hi, everyone. I'm Christina Loiacono, coordinator for the National Animal Health Laboratory Network, or NAHL. And I do really appreciate the opportunity to be able to be part of this whole scientific opportunity here to share the collaborative approach that our U.S. animal diagnostic laboratories are using in response to SARS-CoV-2. To start, I'd like to give you just a little bit of background on the NAHL. The network was established in 2002 as a partnership between two agencies within USDA and the American Association of Veterinary Laboratory Diagnosticians. At that time in 2002, 12 veterinary diagnostic laboratories with expertise already in-house and strong infrastructure already present, these were chosen to take on surveillance testing for diseases of high consequence in animals, such as foot and mouth disease, African swine fever, and avian influenza, just to name a few. The network has grown quite a bit since 2002. Was I lost?
Yes, Christina, hi. Just for a few moments you were.
Okay. Are you able to see my, let me share my slide again.
Please share, yes.
Apologies for that. How's that?
You're back.
Okay. Thank you, and sorry about that. So the purpose of the NAHL is the same today as it was at the very beginning. We provide early detection of high consequence animal diseases through targeted surveillance. We provide rapid response through surge capacity testing should a disease be confirmed in the US. And then we also provide appropriate recovery from a disease outbreak. And that means high testing, high volume testing to provide our trade partners and consumers confidence that we are truly disease free. Okay, so as part of the whole One Health COVID response in the US, the USDA, including NAHL, is part of a One Health federal interagency COVID-19 coordination group, affectionately known as OFIC. This group is led by the CDC, and it includes over 20 federal agencies, each with multiple partners participating in regular calls. And we share information and timely updates, as well as ensure coordination, cooperation, and efficient use, of course, of governmental resources as we jointly investigate and report on our SARS-CoV-2 related activities. So within OFIC, we have 5 subgroups that each focus on a specific aspect of our joint response on the animal side of this pandemic. They include a companion animal group, animal diagnostics and testing, wildlife and zoo, production animals or livestock, and then environmental health. So in representing USDA and NAHL, I co-chair the animal diagnostics and testing subgroup, along with a representative from FDA Center for Veterinary Medicine. And one of the main objectives of this particular subgroup is to monitor animal testing for SARS-CoV-2 in the US. So as far as the NAHL laboratories doing this testing, the NAHL began testing animals for SARS-CoV-2 very early on in the course of the outbreak. Many of the laboratories got PCR up and going very quickly. This map shows a distribution of the NAHL laboratories around the US, and that we currently have 38 NAHL laboratories capable of testing for SARS-CoV-2. And except for in very specific cases where for dogs and cats where a previous dog or cat has been identified and confirmed in a state, all other presumptive positive animal cases from the NAHL laboratories as well as other diagnostic laboratories are sent to NBSL for confirmatory testing. And that's so that USDA can report these cases to the World Organization for Animal Health, or OIE. Now, in addition to the animal testing, and I'm very excited to be able to report this to you, we have 25 of our NAHL laboratories that are currently CLIA certified or the equivalent to test human samples for COVID-19. And these laboratories have completed over 4.3 million human sample tests. And so these are animal diagnostic labs supporting the COVID-19 response in their states and supporting their public health. I think that's just fabulous. Now, as for animal testing, back to animal testing, as I said, we're monitoring the number of animals being tested in the US, and we're reporting this monthly to the OFIC group. The data is voluntarily submitted by the non-laboratories, as well as private laboratories, including Antec, IDEX, Zoetis, and Zoologics, and also by research laboratories doing surveillance testing, and that's primarily in COVID positive households. We request that only testing data from naturally infected animals is included, not including any animals that are intentionally inoculated for research purposes. So on this slide, we're showing the total number of animals that have been tested. So here you can see a hex map of the US, where each state is shown in equal size. It's also a heat map, so the darker the green in the state, the more animals from that state have been reported through our laboratory systems as tested for SARS-CoV-2. As of April 15th of this year, and again, we're reporting out monthly, so as of April 15th, we've had about 7,400 animals tested for SARS-CoV-2 in the US. 27% of those were tested in non-laboratories, 18% in the private laboratories, and then 55% in research facilities. So there is quite a bit of surveillance testing going on in the animal populations. And again, they're primarily associated with COVID positive households. We're grouping animals in 3 categories. The companion animal group includes the highest number of animals tested at 73%. And most of these are dogs and cats, as you can imagine. The wild, captive, and zoo group represents 18%, with bats being tested most often. And then farmed animals or production animals are the 3rd group, with meat being tested most often in that group. On this slide, you can see that the hex map has changed. It's showing the cases confirmed at NBSL by state of origin. It's also a heat map. So the darker the orange, the more animals from that state have been confirmed positive. The total number of confirmed animals in the US as of April 15th is 172. And that's about 2% of the total number of animals tested. It also represents, I think, approximately 61% of the total number of animals globally that have been confirmed positive. The individual animal species and the number confirmed for each is listed on the lower right of this slide. And then the animal species are pictured over on the left. We're not including the otters because, again, this is data through April 15th. So otters were tested and confirmed after that. We're also not including mink in the number of confirmed individual animals, because positive farms are being counted versus the individual animals as far as the mink go. And we do have 16 positive mink farms in the United States currently. And then here on this last slide, you can see the hex map has changed again. And on this map, we're showing the dog and cat cases that have been reported by the diagnostic laboratories as presumptive positive. But they do not require confirmatory testing at USDA, and that's primarily because previously a dog or a cat has been confirmed from that state. So again, we have about 13 animals identified as presumptive positive so far. So we will continue to request that testing information be voluntarily submitted by all the laboratories testing animals for SARS-CoV-2 in the US, and that's so that we can better understand the significance of the virus in these animal populations and how this may affect public health. And that, again, is associated with our One Health approach and our collaborations with CDC, FDA, and many other federal agencies. So if any of you know of testing being done in animals for SARS-CoV-2 and they may not be providing that data to us, it would be really great if you could communicate that to us so that we could contact those laboratories and request that they share that information so we can get a big picture of the testing that's ongoing in the US and animal populations. And with that, I'd like to thank you and happy to answer questions.
Thank you, Christina. Thank you, Mia. Please send in your questions. For the two, we will take them simultaneously. I will ask questions. First one that I have, let's see here. I'm not sure if Jürgen Richt is asking, it looks like it would be, did you try to compare, oops, that means you're supposed to stop speaking now. Did you try to compare CTs for PCR between N and NS12 targets? I'm not sure, I think it's probably to Mia.
I wasn't quite sure I understood the question. We certainly have that data to evaluate. We consider, we're following CDC's testing protocol, and we would consider an animal positive when it's detected by both targets. And we certainly do see a difference, as have others, and that's been described previously. Happy to talk to him directly about more. Thank you.
Very good. Next question comes from Amresh Das, and for Dr. Torchetti. Is there any evidence or report that SARS-CoV-2 could transfer between different animals, either at the zoo or at home? So outside of human, animal-to-animal interface.
Yeah, I think there's the publication on the Brooklyn Zoo lions and tigers is a really good one for you to take a look at. And yes, we do believe, partly based on their behaviors, that there is a great chance of animal-animal to transmission. In households, I think it's a little harder. The vast majority of what we have are known COVID positive households. So did they pass it directly to each other, or are they just being exposed from the human and virus environment? I'm not sure we have a good answer for that.
And of course, experimentally, there have been work demonstrating, at least in ferret populations, the ability to transfer without direct contact or aerosol. I'm aware of those terms as well.
Thank you.
And I'm going to take a moment just to ask, do we know anything about fecal shedding in these species, and whether there's any possibility of transmission just through fecal, either companion animals or other?
Yeah, that still remains a bit of a question. We do know that of the animals we've looked at, there does seem to be kind of later in the course of infection, kind of a little spike in being able to detect it in the feces. So it depends on where that animal is in the course of infection. And it looks like big cats in particular appear to shed for potentially prolonged periods through their feces. Whether that is infectious is a question that, again, kind of goes back to that challenge with virus isolation. Is our virus isolation sensitive enough to really answer that question?
Right.
And I think as we get more tools going forward, we might be able to answer it, but might not have a good answer now.
Thank you. Simon Song asks, wonderful work by the way, he says, from your phylogeny slide, can we assume that the new high pathogenic human strains, such as the D614G ones, are the most infective in most susceptible animals as well? So in other words, is there a correlation between what we see for human infectivity, presumptive infectivity, and the new variants of concern?
There we go. And I think that's an excellent question, and one that we will continue to look at. I'm less, based on the data we actually see, I'm not sure that we can, we could say we know D614G has demonstrated to be more transmissible, and so that's maybe why we're seeing it in animals. But is it a function directly of that, or is it rather a function of what we're seeing in humans that is then spilling over into animals? I will leave it to the solid scientists to go ahead and explore that question a little bit further. From a diagnostic standpoint, are these variants going to be more susceptible in our companion animals and mammals? It's kind of looking that it might be, but again, are we separating out the what's predominantly in those humans as to what we're seeing in the animals? Over.
Just to telescope a few things in our lab, we are looking at ID50 values across some variants. This is work that will be published, we hope, fairly soon, looking at the original Washington 1 strains versus the newer strains. The B117, for instance, we are seeing fairly low levels of virus required for aerosol inhalation infectivity in non-human primates and fairly low in hamster models, very low as a matter of fact. So we will have some comparative data at some point in the future to report. But those are all questions that are swirling. Okay, a question for Christina as well. Can you expand more if samples from animals like gorilla, lions, snow leopards were submitted due to association with clinical signs or due to contact with infected animals or humans or just for surveillance?
Well, based on what the CDC guidance has been, that we at USDA have collaborated on and helped to write, testing of animals is relatively discouraged, right? Because for many reasons, but one of the biggest is we're hoping to restrict the use of resources that could be used on the human testing side. But testing in animals the testing of animals should meet certain criteria and be approved through the state. So the state animal health official and the state public health veterinarians or other officials. Typically animals that are clinical are the first that are chosen. Animals that are unique or endangered species are also on that list. And animals that are group housed are on that list as well. So again, there's a bit of an algorithm that we evaluate the animals against to determine whether testing should occur or not.
Thank you. I'm not sure to whom this question specifically should be directed, but it's from Jürgen Richt. And do you see adaptive mutations in the S protein receptor binding domain region of the mink isolates, similar as reported for this Danish cluster 5 mink isolates? I'm not sure if you have that type of analysis ongoing.
We are looking, we continue to look, and there have been no viruses consistent with what was identified in Denmark, but we do continue to look very, very closely, particularly at the any homoplasia or repeated SNPs, single nucleotide polymorphisms that might occur independently and arise independently in the same species. We're really trying to track and monitor that. So far, we've only associated some of those findings with mink, not in other species, for the reason that there's more of them, it's circulating longer with a much smaller situation in a household or in a zoologic collection. Over.
Thank you so much. Haley, you have to give me an indication as to whether we have a few more questions as to whether we are about to move to a break and then into the panel discussion, or can we ask a few more questions? So I'll rely on you.
Sure, Dr. Korch. Yeah, let's go ahead and take it until 10 after the hour, and then we'll break from there.
Very good. So a couple more quick questions. Chris Owura asks, can we use a standard real-time PCR assay used for the detection of SARS-CoV-2 in humans? Can we use that for dogs and cats?
So great question. And again, you want to consider the virus behavior in the animal. So is the animal going to be infective and shedding from a respiratory sample? So knowing where the animal sheds the virus is very important. Of course, early on, we don't know. We can suggest, we can propose, we can predict based on other similar viruses. So we need the appropriate sample type. But yes, in generally speaking, as long as we've got the appropriate sample type, the tests, PCR tests anyway, used in humans should work on similar samples collected from animals. Where we want to draw that line a little bit, and I don't think he asked this, but just to clarify, serologic tests should be well understood and well tested before we decide to use them confidently in animals, because we have seen where many of the serologic tests developed for humans do not work well in animals at all, or will give you false results.
I'm going to skip down just to another question. And again, I encourage the speakers to go online to the Q&A and answer questions that we did not able to do live directly to the requesters. So this last question comes from Carla Bravo de Rueda. I think it's for Christina primarily. And could you let us know why there are more cases in both Texas and New York compared to the rest of the states, at least on the hex pattern that you showed, the heat map?
Sure, sure. And actually, Massachusetts is the top state right now with the number of animals being reported as tested. That's primarily for those survey studies that are occurring at Texas Animal or TAMU at Tufts in Massachusetts, and other these larger research facilities that are basically advertising for their surveys, doing a great job of getting participation from local households that have COVID positive individuals and are willing to have their animals tested.
Thank you. I want to thank all of our speakers, all 5 speakers, for a truly wonderful set of presentations this morning on emergency response, and Dr. Swain and Chair for starting us off this morning as well, and the importance of emergency response. From my years working in HHS for the Assistant Secretary of Preparedness Response, dealing with human emergency response, all I can say is the vast array of capabilities that you see displayed in the speakers' organizations, as well as those that we haven't presented here, but just the tremendous investments and ability for our government in the U.S. And for other governments around the world to rapidly identify and develop countermeasures or mechanisms for control. I think the challenges are not going to be smaller in the future. They're just going to increase as a function of many different variables. So with that, what we would like to do is take a 15-minute break and come back around, let's see, it looks like 15 minutes. That would be, Christina, you give me a time and I will Haley, give me a time and we'll let everybody know when to be back.
We'll come back at 25 after the hour.
25 after the hour. Thanks, everybody, and hopefully you will be back for that panel presentation.